The prostate is a gland present in the male reproductive system that sits below the bladder and surrounds the urethra. The prostate produces fluid that makes up approximately 30% of the semen, containing enzymes that function to protect and nourish the sperm as well as to alkaline the semen. Prostate cancer occurs when the cells of the prostate mutate, grow and divide to form a cancerous tumour. Prostate cancer remains the third most common cancer in Australia. FOXA2 is a transcription factor that belongs to the fork-head box protein family of transcription factors that regulate gene expression in cell growth, proliferation and differentiation. FOXA2 is only found in the early stages of prostate development and in neuroendocrine small carcinomas as well as late-stage prostate cancer. The increased expression of FOXA2 with neuroendocrine carcinoma suggests that FOXA2 regulates genes which provide tumorigenic epithelial cells with a selective advantage to spread and therefore influence cancer phenotype. Studies have found that under hypoxic conditions, FOXA2 interacts with the hypoxia-inducible factor HIF1A to recruit the coactivating protein P300, which promotes the transcription of various HIF targets including SOX9, HES6 and PLOD2. Co-expression of these HIF targets have been shown to lead to formation of neuroendocrine tumours and lesions in prostate cancer. Studies have also identified up to 40 other genes that are regulated by the interaction of HIF and FOXA2 in transgenic adenocarcinoma of the mouse prostate, which are involved in the regulation of multiple hallmarks of cancer, including angiogenesis and activating invasion and metastasis. This image shows a pluripotent stem cell of a healthy prostate and its progression to a neuroendocrine carcinoma on the left. The red area in the stage labelled B2 displays the presence of FOXA2 in the metastasized tumour.
Studies have indicated that FOXA2 also participates in the SIA-HIF FOXA2 and Wnt FOXA2-CXCR4 pathways, which both result in the successful ability of the cancer to metastasize, particularly to the bones. This is a further comment on how FOXA2 increases the aggressiveness of the cancer and acquires yet another hallmark, activating metastasis. Lastly, it provides the cancer the ability to avoid apoptosis through affecting its interactions with androgen receptors and therefore promotes the resistance of cell death. FOXA2 enables androgen independence. Androgen independence comments on the aggressiveness of the cancer as it is now able to proliferate at an uncontrolled rate and also avoid androgen-induced apoptosis. RNA interference is an endogenous pathway triggered by double-stranded RNA, which silences expressions of genes through a series of biochemical events. dsRNA is processed by DICER, then loaded onto the RNA-inducing silencing complex, RISC. Short interfering siRNA is integrated into the RISC, where the RNA duplex strands is separated and the sense strand removed. RISC uses the antisense strand to guide the cleavage of the messenger RNA, which is mediated by an endonuclease. A lot of trials and research needs to be put into the delivery technique of siRNA. Systemic and intravenous administration is required to treat cancer with siRNA. siRNA are, however, relatively small and have anionic properties and therefore more likely to be eliminated through degradation and renal filtration. Additionally, siRNA does not cross the cell membrane easily and therefore it is difficult to penetrate deep into the tumor for a long enough time. Chemical modification can be applied to the siRNA to increase its stability and also possess safety benefits, however can interrupt the interaction with RNAi. siRNA is rapidly degraded by nucleases in the bloodstream following intravenous administration. siRNA can be condensed with the polycationic vector to overcome this issue. Nanocarriers need to be developed to deliver the siRNA to the site of interest safely and efficiently. The ability of FOXA2 to influence up to 4 hallmarks of cancer and participate in numerous pathways is a comment in itself on the lethalness of this gene and a highlight as to why we should be granted the ability to further develop our drug. The drug is also worthy of development due to its specificity. mRNA regions unique to the FOXA2 protein will be selected to ensure that other FOXA protein members will not be affected, and its method of administration. Intravenous infusion is an effective damage-free administration which allows for almost immediate delivery of the drug to its target. With research, it has come to our attention that FOXA2, despite its significant impact on cancer phenotype, also participates in other physiological mechanisms within the body, which, if rendered inactive, would cause specific side effects. The most prominent is the participation of FOXA2 in multiple pathways, resulting in the appropriate release of insulin. FOXA2 knockout mice showed excessive insulin release in response to amino acids and complete loss of glucose-stimulated insulin secretion. In addition, FOXA2 participates in other mechanisms responsible for the transcription of liver-specific genes, for example albumin. As a team, we recognize the adverse effects and the potential of the drug effectiveness to be maximized. We hope that with the ability to further develop our drug, we may prevent or minimize these side effects, better effectively deliver the drug, and as a whole treat cancer. Thank you.